Last time we just finished talking about the spinal cord and spinal cord injuries. So now we're going to move on to our spinal nerve injury. Now we move on and talk about our last little subsystem of the nervous system, which is the autonomic nervous system. This is a sub-branch of the peripheral nervous system we talked about way back when. And specifically, it's just the motor division of the peripheral nervous system, just the visceral motor division. So as the name sounds, autonomic, of course, it's going to be automatic. So this is going to be the visceral stuff like I mentioned. There's two major subdivisions of this system, the sympathetic and parasympathetic nervous systems, which are your fight and flight response systems for sympathetic, and rest and digest, the opposite, for parasympathetic. The main control of this system is going to be the hypothalamus, although there are a few other areas of it, especially the brain, that have some input. It's mostly the hypothalamus in control of it overall. Hypothalamus, of course, controls homeostasis, which is what the system helps with as well. So the hypothalamus will just kick on one of these two systems, depending on which effect you want to get. As I said, sympathetic is fight or flight. Parasympathetic is the opposite, rest and digest. So between the two, the sympathetic nervous system is the more energy demanding system. You spend a lot more energy when that system's on, because it's preparing you to usually get away or to fight some type of danger. It's active, or most active, during, besides dangerous situations, high stress situations as well. Whereas parasympathetic is going to calm you down from that sympathetic high, and it'll activate mostly digestive and urinary systems, so opposite systems overall. As we're going to see going throughout, almost every single organ is going to receive input from both systems. And if one system turns that organ on, the other one's going to turn it off. Except for blood vessels, they're exactly opposite for each system, depending on which one we're talking about. Another time your fight or flight system is active is during exercise. Always tell students, to your body, there's no such thing as exercise, really. We haven't evolved that much yet. So what do we see with exercise? Your heart rate increases, your breathing rate increases, more blood's going to your skeletal muscles, your body temperature is increasing. That's also the same symptoms you might see during high stress or high anxiety situations. Right, a panic attack, for example, high breathing rate, high heart rate, high blood pressure, all that, because it's the exact same system. The same system is active for both. Because your body thinks you're exercising, you're in danger, really. You're trying to get away from something. Both systems work the same way, in that there are two motor neurons involved for the autonomic pathways. This is different from the somatic motor, your skeletal muscles. They only have one motor neuron from the spinal cord going all the way to your skeletal muscles. The autonomic divisions, they'll have one motor neuron on the spinal cord still, but then they'll talk to a second motor neuron that then goes tell the effectors what to do. In this case, not the skeletal muscles. We call those two neurons, motor neurons, the preganglionic and postganglionic fibers or neurons. And that's because one's before the ganglia we're going to talk about, one's after the ganglia. Look at this image here. On the right would be the somatic system, your voluntary motor system, your skeletal muscle system. We see there's one motor neuron in that anterior horn that goes all the way to your skeletal muscles. On the left, we have the autonomic system. Same thing, we have one motor neuron in the spinal cord, this time in the lateral horn, in this case, of the spinal cord, that then goes to a ganglia, somewhere outside, where the second motor neuron is, that will then go from that ganglia to the effector, muscle, the smooth muscle, the heart, or the glands. So this blue one here that starts in the spinal cord and goes to the ganglia will be the preganglionic neuron, and the purple one from the ganglia to the effectors will be the postganglionic neuron. Both the sympathetic and parasympathetic have this pathway, pre and then post. There are a few differences, mostly in which one's longer than the other. In one, the preganglionic fiber is longer, and the other one, the postganglionic fiber is longer. But they both share the same major pathway. Pre and then the post tells the effector what to do. And a few other small differences too. But any questions so far? No. We're starting with the sympathetic division. Remember, sympathetic is your fight or flight system. It's also called the thoracolumbar system because that's where these neurons are located. I think we should have mentioned the lateral horn of the spinal cord is only in the thoracic and lumbar parts of the spinal cord, and this is why. That's where the sympathetic preganglionic motor neurons are, in the thoracic and lumbar division only. Even then, they're really mostly just the thoracic part. Only L1 and L2 of the lumbar have this system, so it's mostly thoracic, a little bit lumbar too. There are two types of ganglia for the sympathetic system. We have the sympathetic chain ganglia, which are also called the paravertebral ganglia. We call them the sympathetic chain ganglia because they're for the sympathetic nervous system, and they're in a chain. And we can see them here. So on both sides of your spinal cord, you have basically a line of ganglia following the spinal cord on each side. These are the sympathetic chain ganglia. They're also sometimes called the paravertebral ganglia because they're parallel to the vertebra. Now I recommend you only call them, when you learn them, the sympathetic chain ganglia, because a lot of students get confused, think, oh, paravertebral means it's for parasympathetic. It's not. Paravertebral means parallel in that case. Parasympathetic, the para means something else. So if you just worry about learning their sympathetic chain ganglia, you'll remember they're for the sympathetic system and not get confused they're for parasympathetic. But we can see them. So for the sympathetic nervous system, in the lateral horn will be the preganglionic fiber. It will go that short distance to the chain ganglia and then talk to this postganglionic neuron here. That will then go all the way to the effectors. So for the sympathetic division, the preganglionic fiber is very short and the postganglionic fiber is very long because of where the ganglia are located. For sympathetic, the ganglia are right by the spinal cord, so short distance for the preganglionic, and then long distance for the postganglionic. You can see a little bit. There's one more type of ganglia that's used with a few of the sympathetic systems, and that's the collateral ganglia. Somewhere here. There we go. Collateral ganglia. These are located a little bit further away than the chain ganglia, but they're still not all the way by the organs. They're probably about halfway between the spinal cord and the organ they go to. So in that case, the preganglionic is a little bit longer, kind of equal in length to the postganglionic, but it's not the opposite of what we see in the parasympathetic. So for sympathetic, there's two ganglia types. There's the chain ganglia right by the spinal cord, which are used by most of the system. And then there's the collateral ganglia, which would be like this ganglia here, a little bit further away than the chain ganglia are. Also, doesn't matter which ganglia we're going to, it's still the same pathway, pre to post, and then post goes tells the effector what to do. Which also is this. There is one exception to the pre-tell post, tells the effector what to do. And that's when the sympathetic system activates your adrenal glands, specifically your adrenal medulla, which you'll learn more in AP2. The medulla of the adrenal glands is what makes epinephrine, norepinephrine as hormones. Well, remember, this is the fight or flight system. Your fight or flight hormone is epinephrine and norepinephrine. So we want to activate that medulla as fast as we can, because between the two systems, sympathetic and parasympathetic, the sympathetic is more important that it's fast, right? If you are immediately in danger, you got to kick on very, very quickly, or else you might die. Whereas if you're done and you're trying to rest, you can take longer to calm down because the danger is gone now. So to make the sympathetic system faster when activating the epinephrine, norepinephrine hormones, there's only one motor neuron involved there. There's only the preganglionic fiber going to the adrenal medulla directly, which we can see in this image. So as you see, whether it's the chain ganglia or the collateral ganglia, it's always preganglionic in the spinal cord, talking to the postganglionic in the collateral ganglia, that then goes to the organ, the effectors to have some effect. But if we look down here, right above the kidneys, that's your adrenal glands, you'll notice there's just the blue there. And that's because this blue one starts in the spinal cord and it's going to go all the way, activate the adrenal medulla directly. So between the two systems, para and sympathetic, that's the only exception. For the sympathetic activation of your adrenal glands, it's just the preganglionic fiber. To make it faster, one neuron's going to be faster than if two neurons are working together. Questions on the sympathetic system overall? The opposite of it, in almost every way, is the parasympathetic. Sympathetic was thoracolumbar. Opposite of that would be craniosacral. So parasympathetic is the craniosacral division because that's where those motor neurons are located. Most of them are in the cranial part, in which case they're from that vagus nerve we learned about previously, number 10, the 10th cranial nerve. There's a little bit from the sacral part of the spinal cord, but about 75% of the entire parasympathetic system is going to be controlled by the vagus nerve. A few of the other cranial nerves help with it. 3, 7, and 9 help a little bit. But again, it's mostly the vagus nerve in charge. Its ganglia are called terminal ganglia because they're at the very end of the pathway. The terminal ganglia are either right outside the organ or inside the organ, which you can see here. So mostly for the organs around the skull, they'll have terminal ganglia on the outside. Most of the other visceral organs, the ganglia are actually inside the organ they're going to. So in this case, again, we have the opposite of sympathetic. The preganglionic's very, very long, and the postganglionic extremely short. So again, the opposite of the sympathetic blitz. Question on the parasympathetic system. So what effect each system has is going to depend on which organ we're talking about. A lot of people get into their head that sympathetic excites everything and parasympathetic inhibits everything, and that's not true. They both are excitatory and inhibitory depending on the organ, because depending on what we want to see. If you want to fight or flight, you want increased heart rate, increased breathing rate, increased blood to your skeletal muscles, all that. But you don't want to be digesting food then. So sympathetic is excitatory for heart, lungs, and skeletal muscle activity, but it's inhibitory for digestive and urinary system. Parasympathetic is the opposite. It's rest and digest, so it's going to activate your digestive and urinary organs, but it'll inhibit or slow down your heart, your breathing. So both systems do both, just depends on what organ we're talking about. If you exercise, you kind of see this, if you're to think about it. Again, you're exercising, so of course we got increased heart rate, increased breathing rate, that sympathetic system. Well, if you exercise for like 2-3 hours at a time, you might notice during that time, you don't even have to go to the bathroom at all. That's because the sympathetic system is turning off digestion and urinary. Because again, think about it as you're in danger. If you're trying to run away from danger or fight danger, you don't want to go to the bathroom at that point. You want to fight the thing, and then when you're resting later, when para turns on now you can afford to go to the bathroom. So they're both excitatory and inhibitory. Just depends on the organ we're talking about, which one does what. Another difference between the two systems is which neurotransmitter which fiber releases. Some of them are cholinergic, meaning they release acetylcholine, and some of them are adrenergic, meaning they release norepinephrine. The adrenergic there comes from adrenal, because norepinephrine is also released by the adrenal gland, so adrenergic. As I mentioned before, though, norepinephrine is both a hormone and a neurotransmitter. Epinephrine is only a hormone. So adrenergic neurons only release norepinephrine, not epinephrine. And this terminology is not just used here. If any neuron that releases acetylcholine would be a cholinergic neuron. Anyone that releases norepinephrine is an adrenergic one, but that's what we talk about here. But for example, we learned your skeletal muscles always contract because of acetylcholine. So your somatic motor neurons are also cholinergic. In this chart here, we can see which fibers are which one. On the top, we have the parasympathetic division. What would that preganglionic neuron of the parasympathetic be? That top one there. Would it be cholinergic or adrenergic? That blue one on top. What's it releasing? It's releasing acetylcholine, right? So it would be cholinergic because it releases acetylcholine, right? Cholinergic acetylcholine. If you look at the postganglionic for that one too, it also releases acetylcholine. So for the parasympathetic system, both neurons are cholinergic. The preganglionic is cholinergic, and the postganglionic is cholinergic for the parasympathetic system. They're both releasing acetylcholine. Look at the bottom two, which are the sympathetic systems. You notice again the preganglionic is cholinergic, but now the postganglionic is adrenergic because it's releasing norepinephrine. Remember, it's really the postganglionic neuron that's the important one, right? It's the one telling the effectors what to do. So they're the ones that would need to be different to have those opposite effects. The reason why sympathetic speeds up the heart is because it's releasing norepinephrine onto the heart. The reason why parasympathetic slows down the heart is because it's releasing acetylcholine onto the heart. Different neurotransmitters are going to have different effects on the same organ. So of the four, the only one that's adrenergic is the postganglionic sympathetic. The others are cholinergic. Want to remember which one's adrenergic is think, okay, sympathetic's fight or flight. What are the fight or flight hormones? Epinephrine, norepinephrine. So the postganglionic sympathetic has to release norepinephrine because that's the adrenaline one. That's the fight or flight hormone. You'll also notice this image also shows you the length of the two fibers. Again, the preganglionic para is long. They have the terminal ganglia with short post. The sympathetic has shorter preganglionics, longer postganglionics, and they have the chain or the collateral. Questions on the types of motor neurons each system has. So we see that the reason why each system has opposite effects on the same organ is because they're releasing different neurotransmitters. Again, the sympathetic speeds up the heart because it releases norepinephrine. Para slows down because of acetylcholine. But why does norepinephrine speed up or activate every organ? Again, the sympathetic system is going to slow down digestion because norepinephrine slows down digestion and acetylcholine speeds it up. How though? How can a neurotransmitter have different effects on different organs? Why are they always excitatory or always inhibitory? We talked about previously when it comes to other things. What's really the important thing that determines what the organ does? It's not the neurotransmitter, it's the what? So it's the receptor that determines the thing. So the reason why norepinephrine will speed up the heart but slow down digestion, it's because the heart and your intestines have different receptors for norepinephrine. The heart has the receptor that speeds up with norepinephrine. The intestines have the receptor that slows down with norepinephrine. The neurotransmitter does matter because that's what the receptor is looking for, of course, but it's the receptor that determines the effect. There's a lot of different receptors for basically every single neurotransmitter, hormone, really anything you're talking about. We're going to talk about two for each time. So there's two main cholinergic receptors. So these two receptors both will bind to acetylcholine, cholinergic, which I mentioned them previously, the nicotinic receptor and the muscarinic receptor, named because the nicotinic also binds nicotine. The muscarinic also binds muscarine, which is a type of fungal toxin. Again, that's not what these receptors are supposed to bind. They're supposed to bind acetylcholine. It just so happens those two, nicotine and muscarine, look a lot like acetylcholine at their certain parts. So for these two, and there's other types as well, these are the two big ones. They're both always excitatory. It's just the speed of them is different. The muscarinic receptor is always excitatory, but always slow, relatively slow. The nicotinic receptor is always excitatory and always fast, very rapid. And you kind of see that if you know someone who is basically addicted to nicotine, they get the withdrawals, but basically as soon as that nicotine gets into them, they already start to feel better. That's because nicotine binds to the nicotinic receptor, which is very, very fast acting. That's why nicotine patches, basically the first drag of the cigarette, makes them feel better almost immediately, because it's a fast receptor, that nicotinic receptor. These are more of the fight or flight receptors, the nicotine receptors are. When it comes to adrenergic receptors, there are two types we call alpha and beta. There's a lot of subtypes of each of these as well. I think there's like 12 different types of beta receptors when it comes to epinephrine, norepinephrine. Yeah. Depending on, they're the one that's kind of more varied than the acetylcholine receptors are. Which subtype of alpha or beta they are will determine if it's excitatory or inhibitory, if it's fast or if it's slow. Doesn't get all lost up there. As I mentioned previously, and you'll get this more in AP2 though, but I might ask a question about it here. Remember, norepinephrine, epinephrine, there's no difference. They are technically different chemical, yes, but they have the same effects everywhere. Norepinephrine, epinephrine, they're both basically adrenaline. But norepinephrine is both a hormone and a neurotransmitter. Epinephrine, just a hormone. Questions on the receptors? So lastly, let's talk about how we stop these effects. Yes, we want sympathetic to be fast and last for kind of a long time, but we eventually want to stop it. And same thing's true for parasympathetic. We don't always want to be relaxing. We want to turn that system off too. So remember how we get rid of neurotransmitter, or how we stop a signal? We have to get rid of the neurotransmitter. Well, for the cholinergic neurons, we already know how that works because we talked about them with muscle contractions. To break down acetylcholine, we use acetylcholinesterase. So just like your skeletal muscles had acetylcholinesterase at the synapse, everywhere acetylcholine gets released to, they also have acetylcholinesterase to break down the acetylcholine there. So acetylcholine gets broken down inside the synapse by acetylcholinesterase. Norepinephrine is a different class of neurotransmitter altogether. Norepinephrine is one type of monoamine neurotransmitter we mentioned before. Monoamine just means it's a modified amino acid. It's very, very simple pretty much. For monoamines, they get taken out of the synapse through a process called reuptake. So if you reuptake something, you basically suck it back in. So for the adrenergic neurons, they release the norepinephrine and then they suck it back up into them. And that's reuptake. Once they're back in the neuron, they then get broken down by monoamine oxidases because they use oxygen oxidase to break down the monoamines. So where the signal stops basically is different. For the adrenergic neurons, the signal is broken down in the synapse. What did I just say? For cholinergic neurons, the signal is stopped inside the synapse by breaking down acetylcholine. For adrenergic neurons, the signal is stopped by sucking it, vacuuming back up the norepinephrine. We then break it down, but it stops because we suck the norepinephrine back up into the neurons. That kind of plays a bigger role when we're looking at, okay, what if we want a drug to affect how long the signal lasts? Like let's say we want the cholinergic or the adrenergic signal to last longer. How would we make the cholinergic signal last longer? Well, we need acetylcholine to not get broken down, right? So we need to find a drug that stops acetylcholinesterase. Because if that stops, it's no longer broken down. It stays in the synapse longer, so the effect is longer. So you would find an acetylcholinesterase inhibitor. For the monoamines, like norepinephrine, we can't, or what works is not stopping them from being broken down. Because the signals stop before they're broken down, right? We reuptake them first, then break them down. It's the reuptake that stops the signal. So we need to find something that stops reuptake of monoamines, not stop them breaking down, but stops the reuptake. Those are drugs we call reuptake inhibitors. So SSRIs, NERIs, those are serotonin and norepinephrine reuptake inhibitors. questions on termination of these signals? So, like I said, the control of these two systems is mainly the job of the hypothalamus. It's looking at homeostasis. If it's messed up, it'll turn one of the systems on, the other off, depending on which way we gotta go to fix it. Because again, one is excitatory for some things and inhibitory for others. There are some other parts that also affect the system, especially the breathing and heart rate systems. We learned those were in the medulla. So medulla has a little more input when it comes to the cardiac speed and the respiratory speed. But mostly everything else is the hypothalamus. Another system has another system that can influence the autonomic systems is your limbic system. Limbic system we mentioned here and there is your emotional system, your emotional control system, which we've all experienced this problem. If you wanted to right now, you can basically make yourself have a panic attack by just thinking about very anxious things, getting yourself hyped up, emotional. You're basically activating your sympathetic system, indirectly activating it, but you're influencing it still. You can also activate or influence your parasympathetic with your emotions as well. If you're currently very anxious and having a panic attack, just trying to start to get calmer will activate your parasympathetic system to help you rest and calm down. So your emotional state can also influence this system. Doesn't control it, that's the hypothalamus. But you can influence it by basically your mindset and the various breathing techniques or whatever, depending on which way you want to go. Lastly, let's look at just lifespan changes for the nervous system in general. The lifespan changes here mimic the lifespan changes we see anywhere in the body, as I always say. As you get older, you start to have less cells involved, so we do see decrease in functioning of the nervous system as you get older. But it's also changing from the very beginning. During development, during the fetal stages, your brain, in terms of neuron numbers, is very large. You have more neurons before you're born in your brain than you do by the time you're born and for the rest of your life, which is bad actually. You have too many neurons in your cortex during fetal development. You need to get rid of some of them because you have so much, it causes overactivation. And we see that in some people with early onset, like childhood schizophrenia, they have the same number of neurons as a fetus would. So that's too many neurons causing the hallucinations and other things we contribute to schizophrenia. It's just too many neurons. It's too much activation of the brain. So you want some of those neurons to die, and that's the process of apoptosis we talked about way back in chapter 3 or 4. Over time, after that, your brain does shrink a little bit. Like once it stops growing, it then shrinks a little bit, about 10% throughout your entire life. That does not necessarily immediately represent how many neurons that are lost. You actually lose from basically now until you're 90. You'll have lost half of all the neurons in your cortex, but the brain only shrinks by 10%. Remember, it's not just neurons in your brain. Most of the things in your brain is actually all the neuroglial cells, and they remain pretty constant throughout your life. Another interesting thing is that where these neurons are lost is not equal. We see more loss of gray matter compared to white matter. So you lose more of the processing part of your brain than you do the signal sending part, the white matter. Where they die off is also different. You lose more neurons in your temporal lobe than you do in your brainstem. Which, remember what these two parts do, that makes sense, right? Your temporal lobe compared to your brainstem is not that important. Temporal lobe does a lot of sensory stuff, especially hearing, Wernicke's areas there. Broca's overlaps a little bit with it, etc. Olfactories here. So you do lose some of your sense processing as you get older because your temporal lobe neurons are going away. But your brain's neurons are fine, which is why your heart always still beats. Your respiratory rate's still fine. The life-sustaining parts, we don't want to lose stuff in, so we try not to. Yeah. One thing we see as well is that even though, if you get to 90, you have only half the neurons in your brain you did when you're 25, we don't always see a reduce in function in some people at all. Really, unless someone develops a neurodegenerative disease like Alzheimer's or dementia, we don't see that much of a decrease in function compared to when they were younger. Part of that's because, again, it's not always the neuron number that's important, it's the connection number that's important. So some neurons will compensate by making more connections. Now, we do see a loss of connections overall, though, as you age, which does lead to that slight decrease in functioning, but it's not the half function you would expect from losing half the neurons. Other things we see less levels of neurotransmitters as you get older, meaning it takes longer to reach threshold because those less neurotransmitters are released. We also see the action potential itself is slower. It slows down by about 5 to 10%. So kind of all that together, less neurotransmitters, longer to reach threshold. When it does, the action potential is slower. All that accompanied with stiffer joints, slower muscles, weaker muscles, leads to all the increased falls we see and slower reflexes in the elderly. As we mentioned in sleep as well, but yes, older people go to bed earlier, they wake up earlier, but remember, they're not sleeping the whole night. I think it's like half to 2/3 of everyone over the age of 60 experience geriatric insomnia, where they might sleep for 2 hours and then wake up for 2 or 3 hours, then go another 1-2 hours of sleep, and that's not a healthy sleep cycle. So we do see a lot of insomnia in the elderly. Mostly because of the parts of the brain that control sleep breaking down, getting weaker, less melatonin getting released, all the stuff you kind of would expect. Kind of going back to sleep, I watched a sleep video the other day. We talked about the concept of sleep debt, right? If you don't get a good night's sleep, you make up for it the next night. Well, research actually showed that pulling an all-nighter actually takes your brain a whole week, 7 days to completely recover. So it's not just the next day you feel bad, your brain feels bad basically for a week if you don't get a single good night's, like a really bad night's sleep. So try to sleep the best you can. And the all-nighters are not worth it. That's it for chapter 11. Let's do the essay question. Everyone want to come do it for bonus points? Have you done it for this one yet?
Yeah.
Make sure it's working. Yeah. And use this side. So my question while she gets this started, starting from resting memory potential, explain the process of an action potential. We should list off the millivolts, the names of each segment, and then what's happening in each segment. So again, just explain everything about an action potential.
Press is -70 millivolts. Local potential, bring it to threshold at -55 millivolts. And the sodium channels open
What kind? What kind of sodium channels?
Voltage gated.
Voltage gated sodium channels.
Voltage gated sodium channels open, allowing sodium to come in, which makes the inside positive. Polarization. Can I just put depot?
Depot is fine.
Okay, until about positive 30 millivolts. That's when the potassium channels, voltage gated channels open and the sodium channels close. And this called repolarization. And the potassium channel is close too slow, which causes the hyperpolarization at brings it to negative 75 millivolts. And then the potassium and sodium pump opens,
I'd say turns on.
or
Activates better. Say open, that's fine.
pumping the sodium back out and the potassium back in, bringing it back to rest at negative 70 millivolts.
I don't remember, I heard you, if you said or not, if you said the potassium leaves, you didn't say it's going to say that in the explanation. I don't know if you said that.
Leaves whenever it leaves the inside.
Well, when the potassium is open, you said that. I didn't hear if you said which causes potassium to leave the cell. If you did, just make sure you put it down there. Like don't just say the channel's open. What happens now the channel's open? So we'll go in, that's good. This real quick as well. So yeah, we start at rest, which is negative 7 millivolts. Nothing will happen until we get to the threshold, which is negative 55 millivolts. There, the voltage gated sodium channels open, so sodium will come in now, depolarizing us up to positive 30 millivolts. There, the voltage gated sodium channels close, those voltage gated potassium channels open. Now potassium will leave this cell, causing us to get negative, which is repolarization. But too much potassium leaves because those channels are slow to close, they're lazy, hyperpolarizing us down to about negative 75 millivolts. To get back to rest, the sodium potassium pump sodium out, potassium in, to reset us to negative 7. And then we'll run another reactor potential, another one a few times. Any questions on the essay question? So we're not going to start chapter 12 at all. I don't like doing it, honestly. But that I told y'all to read chapter 12, just so we technically covered it. And if you're interested in like going to optometry or like ear, nose, throat, especially like ear stuff, as a nurse, that's the chapter where it covers a lot of the stuff in it. So I do recommend if you're interested to read over it. This not be a chapter 12 quiz though. Chapter 12 will not bring your exam at all because we might cover 10 slides. It's not enough to use on. But your exam, which will just be on chapters 10 and 11, is going to be on Thursday. Normal exam, like we said. Does anybody know they're going to miss exam 4? You good? So, yeah. But anybody else might miss exam 4? And what should be good? You don't have time to make it up. Wednesday, Thursday, but my 2 classes. Then Monday, Tuesday, that's fine. That's it. Your final will be on Tuesday next week. If you can't make that, you can make Monday's class instead. Same time, same place, same exam. But there's no other time to make it up. It's crazy Wednesday, so. The final is cumulative. The questions are going to come from the past exams. Besides that, though, it's just like a normal exam. You'll have one essay question, one of the 4 we've done before. I'll pick one of them, and it'll be mostly multiple choice. Probably a few matching sections, smaller matching, but maybe multiple matching sections to cover multiple exams. But it's a normal exam except everything. Same length, same amount of points, all, it's the same. So Thursday, normal exam, so I'm sure most people finish about an hour, like normal. When everyone's done, we'll come back in. I'll review exam 3. I should have exam 4 graded by then as well, and get back to review exam 4 as well. So you take pictures of it and then use it to study for the file if you want to take it. The file is optional, you don't have to take it. It'll only bring your grade up. If you do bad, it won't bring your grade down. Your lowest exam drops, if that's the final drops. If you don't take the final, your final is your lowest exam then because you got a 0, so the final drops. You have to, if you want your other exams to drop, you have to take the final. You do better. If you do worse, the final just goes away. Since, like I said, I'll grade exam 4 that day. I should have time to start grading it while I'm taking them, and then I might be like, hey, well, I'll give me a 15-minute break. Y'all can come back in. I'll finish grading that 15 minutes. I'll put your grades in the week you want. If you have time, which you should. One time y'all can come up, see your grades in the class overall. I can tell you, make this on the final. This is the grade you'll have. If you need bonus points, do that. That'll help too. Blah blah blah. Okay, and I can tell you if the final will even help you or not to move to the next level. Any questions about the process of the last week?
Okay, so exam is Thursday, not the final exam next
Tuesday, yeah.
Should the bonus points be put in by Thursday so we can see for sure?
If I can get to them, yeah. If I don't grade them officially, I can put them in, assuming you do them correctly, and then so we got to figure out what those ones are.
And for the survey, we just screenshot it and just email it to you?
Yeah, so
or-
don't screenshot your answers, screenshot the green check mark.
So it wasn't uploaded. Like when I clicked on it to do
Last
it, yeah, mine's been loaded. It's not uploaded.
Have you already done it?
No,
They're trying to do it. Like it would pull up at all.
it just shows something, it was an error.
Try it tonight. There might have been something wrong with the stuff.
Well, I just did
Like today.
that.
Might have been something wrong with the system last night. Try it tonight. If it doesn't work, let me know. It's supposed to be open till next Tuesday, so it's still open for now.
Is there another way we can listen to the recordings? Because I'm still not able to.
I'm not have time to listen to those, unfortunately. So. That's why I do like YouTube videos if you want to watch those. But I don't have time Any other questions about like the process of the last week, or bonus points or anything I'm trying to do? Yeah. We'll do exam 3 and exam 4 review on Thursday. We have an hour left still, so next hour we can just review for exam 4 in general. If you have any questions about from chapter 11, let me know. We can go. Oh, don't forget your quizzes are due, like always, class time on. I'll need to remember I dropped up to your 2 lowest quizzes, so I'll be doing that as well, and I'll come up with a good grades. I have to go in and drop them myself. They don't drop automatically, so I gotta wait till you actually do them too. And same thing's true for life. You know what to go.
Can we go over something that was on one of the quizzes? It's just one question. Yeah. It's the
I told you I
somatic,
went and fixed one of the questions in the last quiz. There's another question. I think it's in this actually. It asks about this is probably one you're asking about like which one of the cranial doesn't exist, the special, general, special thing. I forgot to take that one out and change it with a different question, so ignore that question for now. It's like special, general, afferent, special, general, efferent.
Special somatic, is
Yeah,
that the one?
so just put whatever answer you want for that one for now. Once they're due and turned in, I'll go back and give on points for that question. It should be, I think, chapter 11. I forgot to change that one out.
Okay, so that's the one I was.
That was one, yeah.
And then, is your brain, functionally bilateral? That's true or false?
It's true, false.
I put true and it said false.
It's not functional bilateral.
No.
Remember, structurally, physically, looking at it, it is bilateral structurally. But Broca's and Wernicke's are only on the left side, right? But not on the right side, so that makes it functioning not bilateral. That's just one example. Your brain's basically completely functionally not bilateral.
On the study guide for chapter 11, you have organization of the cerebrum. You want just the structure and the function, or?
So, but I think what I meant by that was the organization is like brain is gray matter outside, white matter inside. Spinal cord is the opposite. Except for what part of the brain is not gray matter outside, white matter inside. It's the opposite. It's white matter outside, gray matter inside. Spinal cord, and there's one part of the brain that's also like that. So it's the medulla. The very end, that becomes the spinal cord. It's more like the spinal cord than the brain. It's white matter outside, gray matter inside. So the medulla and spinal cord, the brain in general is gray matter out, white matter.
Okay.
And peripheral system is white matter and gray matter, white matter out. Just the brain that's opposite.
So the matching is just the cranial versus the function.
I guarantee that'll be one of the matchings, yeah. I can't think of, like, I don't remember everything we talked about, so there might be other matching too. I can pretty much guarantee that will be matching. If not matching, definitely about to tell me the functions of a lot of the cranial nerves. Probably matching. But less push out there.